Rare diseases target a small percentage of the general population. Most of these diseases are chronic and serious. There is a lack of scientific knowledge and medical care. Thus, the diagnosis of rare diseases is challenging and therapeutic options are scarce. Rare diseases include a group of around 30 vascular anomalies, which are defined as localized defects in blood or lymphatic vessels. Vascular anomalies are divided into two categories: vascular tumors and vascular malformations. VA-Cure project aims to uncover causes and mechanisms and novel therapeutic strategies for these vascular anomalies. VA-Cure project brought together 7 leading academic research groups, 2 companies and 9 partner organizations from 5 different countries across Europe. In addition, 14 international PhD students were recruited for the research. Students will carry out cutting-edge research with state-of-art techniques. Students will receive intensive training from experts in vascular biology and will take part in workshops that improve their research and transferable skills. Students will have intersectoral visits within our network in order to benefit from each other's facilities and experience. Together, we aim to cover the whole innovation chain, from patient to bench and back to the patient. The VA-Cure project is divided into 4 phases. In the first phase, we will use biological samples from patients in order to find novel, commonly affected genes which might be key drivers of vascular malformation development. The second phase focuses on defining molecular mechanisms of vascular anomalies in vitro VA models and ex vivo samples. Using cell culture approaches, we gain insight into the effect of the newly identified gene mutations on single cells. To validate and further understand molecular mechanisms, we use zebrafish and mouse models. Moreover, fish develop fast, have numerous offsprings, and their genes can be altered with relative ease. As a mammalian species, observation in mouse models for vascular malformation will highly resemble the disease mechanisms in humans. Final phase is the preclinical testing of drugs. In this phase, we will validate diagnostic biomarkers for vascular anomalies and to identify and test potential molecular therapies.